Host. Okay, yeah, so today we have two topics. First, we'll start with talking more about grid cells and location representations and how that could work in the neocortex. Jeff and Hojae have been doing more of a deep dive into the literature there. And then, depending on how long that takes, I think Niels, you prepared a bit also on attention. I don't know if Scott, if you also had any more things on that that we can talk through. All right, then. Yeah, Jeff and Jose, I don't know if you have any slides or things or just brief form discussion.
Well, I think Hojae, you've got some stuff. I spent the last hour trying to get my internet to work, which started working about 5 minutes ago. So all the things I was going to write up this morning, I have very few of them written up, but we can still hack it. I don't know what's going on with my internet. Um, I had to reboot my machine and restart everything. I couldn't get into Excalibur. I don't know. Um, but I have some overall notes, and Vivian and I looked at some papers, and she was going to do a little bit more explanation of what those papers said and the networks involved. So I don't know, Hoja, if you want to start with that, or should I give a preamble of describing what the overall task is we're trying to do?
Um, yeah, I think your
Oh,
preamble sounds good because I was just going to go into details about the two papers that we talked about yesterday and kind of
Yeah.
and yeah, I
go do a
didn't.
comparison if it's needed. Yeah. Sorry. Go ahead,
Yeah, and
Vivian.
I don't know, Jeff, if you thought I looked at that paper. I didn't look at that one paper because I saw that Jose posted some replies to your questions on Slack
Right,
already.
right, right.
So I
I
didn't
didn't.
have a chance to look at that. I looked at a couple of other papers on grid cells in the neocortex today, if
Yeah.
that comes up. But
Well, that would be helpful
yeah.
because I mean, I spent some time yesterday and early this morning reading some papers. Um, but as I said, didn't get a chance to write my notes down. Um, but I'll just give it the overall picture. Um, and I have a couple of, I'll have some notes on in Excalibur, very, very little. So just to remember, maybe I should just go to Excalibur right now. I'll share my screen. Hopefully you see that.
Yep.
And hopefully you see this.
Yep.
All right. So last week I showed these challenges we're working on. And these are all things related to reference frames. And number one here again is shared learning. Um, it's clear that at times that multiple columns have to be sort of lockstep at the same time. They must be observing, I mean, even if they're not observing the same thing, they have to be if they're going to learn, if we're going to do shared learning, that one column's getting the input, the other column's going to learn it. They have to sort of lock onto the same reference frame, or at least reference frame representing the same objects. Um, and they have to, as I say down here, requires multiple columns anchoring to a reference frame for the same object and sharing movement information feature data. And just this is just one of several problems related to reference frames, and it's really challenging. And then it made us think like, okay, we never really got our reference frames working properly, in at least our model in the biological model. Again, this is all neuroscience. And in the biological model, this is how I'd been viewing it. It's sort of I didn't think it was completely right, but it was the best I had. And in this case, we had two cellular layers, layer 4 and layer 6A. Layer 6A representing the location, and we had the concept in layer 4 that you have the mini columns represent basic features, and then you pick for each basic feature, you can pick a cell out of the mini column, and you get an SDR, a unique representation of that feature. So I'm not going to go through any more than that. And we had a similar idea going on in layer 6A, where I said, okay, well, we have a grid cell module, and the grid cell module has, a few cells that are sort of bump of activity, which don't really represent a very high resolution area. I mean that these cells repeat over and over again, and so that you can't really say where you are based on them. But if you picked a cell and if there's a mini column associated with each grid cell, then you'd have a unique representation. Um, of that location in the context of an object. So this is how I was viewing it in the biology. We don't do it this way in Monty. This idea was simple and clean, but it had some real problems. It turns out we don't really know how grid cells work for a lot of ways. So this was a guess that how this might be going on in the cortex. And one of the big problems of this one, this idea that we have, oh, there's a grid cell module, and there's each grid cell there's a mini column, is that the grid cell modules we know can do path integration. So when you have a movement vector comes in, the bump moves around to reflect where you are in the environment. But we had no way of really doing path integration on the SDRs, that is on these cells down in the mini columns. We hypothesized several different ways, but none of them really worked well. And so there was a number of problems with this idea. It was simple, it was clean, it was a way to put it aside for the moment, but it doesn't really work. And we should know how to get it to work. And so, so this was just where we were, and given some of the problems we're trying to solve related to reference frames, we went back and started looking at grid cells and place cells again. And I think one of the this little block down here um, is basically this is my notes for today. So all I had time to write in the last 5 minutes. So um, uh, I think there's some real insights I didn't understand. A lot the grid cell and place cell literature is been going on for a long time, and there's still a lot of things that are being learned. And I haven't been into it recently, so I'm picking it up, and I'm feeling quite behind on it. But here's a couple of things that I thought were really interesting that and these networks that Hojae is going to talk about are implementing this, showing that this stuff works. First of all, I didn't quite understand this, but in detail, but grid cells and place cells are initially determined by environmental clues in a deterministic model-free way. So if an animal walks into an environment, um, there's immediately a set of place cells, and and the grid cells anchor. And this is done not in a random way, but based on large environmental clues, the shape of the room, large features in the room, less so about local features in the room. So you imagine a rat going around a box, and it creates a map of this environment right away, and it's doing it based on large and distal clues. If there's the shape of the room, there's a big objects on the walls, less so by smaller items in the room. So if there's some toys in the room, those don't really anchor the grid cells, and they don't anchor they don't determine the place cells. So this is a really this has been known for some time, and I just didn't understand the implications of it. But essentially, it's saying that grid cells, if you think of them as the map of the room, are determined in a model-free way. That is, this room hasn't been learned yet, and the animal walks in there and immediately establishes a reference frame that it's going to continue to use for that room. And that reference frame is determined by large-scale clues. It isn't learned, so it's in a deterministic way, and so that's pretty interesting. And as I said, the overall room shape and largeness of clues primarily determine the mapping of grid cells and place cells. If something like this occurs in the cortex, so imagine we have something like this going on in the cortex, it implies that reference frames are determined by and represent large-scale features in a model-free way. The overall shape of an object determines its reference frame, and it's not learned, which is not how I've been thinking about it. I've
Yeah,
had
I guess at least the initial reference frame, right? Like sort of
right,
almost like the sort of space that the learning is going to take place in.
right. But and you can look at it, and so what happens with the animal as the um. All right, we're back to where we were, I think. Um, if I can see my screen.
Yep.
Uh, we were just talking, Niels, you were just saying something about um yeah,
I
we
was
were talking about
just trying to clarify because maybe to someone listening, it could be confusing when we say like the shape or the of the reference frame or whatever. Like the reference frame is the space in which a shape could be learned, but you can
well, right,
also pick different spaces, as in like different alignments of the grid cells, kind of put you in different encoding spaces.
right. But
And what you're
what
saying
I
here is the general
what
shape of the object could inform that initial
right,
sort of
that's what it looks
seed
right.
almost.
That's you're right. It's that'd be more clear. Thank you. Right. It's the idea that the general shape of the object determines the reference frame. Um, and It's more like, which is different than I've thought. We've always thought that, reference names are like blank slates and you'd start filling them with information. But
But
this
it's
impl
still like it would still you can still have rooms of the same shape, but connected by a hallway or something. But you would still use different anchorings for the two rooms, right?
well, no, that's an experiment that was actually done. It's very interesting. If they have two rooms that are connected together, initially, and the two rooms look identical, initially the rat will see them as the same. The grid cells will anchor the same way, so the rat can't tell, which room it's in some sense.
I mean, if they have some differences, but they have the same shape, like they have some
Right.
different features
Right.
in them.
It's not just shape. It's distal features, I guess, is what the term is often used, which includes walls and things on the walls. This is a typical rat environment. The point
Because
is that it is something about the overall environment determines the anchoring of the grid cells, um, and the placement of the place cells. And if you rearrange the large scale features of the environment, you get a different mapping.
yeah, and
And if
like
you
maybe
had
one way of thinking of it is, I guess, like if there was a small change, it would be more likely that okay, this small object has just appeared or moved in the same room. But if there's
right,
a more significant change, it's like that implies it's a totally different, yeah,
right.
different room.
The way I view it is I imagine you could be the rat and you would say, okay, do I think I'm in the same room or do I not think I'm in the same room? And so what determines what you think you're in the same room? If I go in my dining room and there's an extra chair, I don't think I'm in a different room. However, if the walls are all different, I would think I'm in a different room. And there's lots of ways this could happen. There's lots of experiments where an environment, a room for the rat, and they move one of the walls in a little bit. They start changing the shape slowly, and initially the grid cells start compressing, like it still wants to map into that room. It's like, yeah, it's the same room, but just I'm just going to accommodate. And at some point it breaks, and then says, no, this is different, and you get a whole new remapping. It's also there was an experiment. I might just say right here. You can have these two rooms that are connected by a door or a passageway, and the two rooms are identical. They have some features that determine what they look like. And initially, when the rat goes from room A to room B, it maps them the same way. It sees them as essentially the same room. But if the rat then becomes aware that these are connected, and like they open up the door a little bit to make it wider, then all of a sudden the rat it'll flip, and one of the rooms will be mapped on the original room, so that it'll become one map for both rooms. And so it's like, oh, now I'm in this sort of larger connected area. I'm going to map it as one space, as opposed to two different rooms that look identical. So these are all the
Yeah,
influence.
and
I think. Go ahead, Mills.
I was going to say from the to the kind of related to the conversation from last week, you could imagine how sort of a certain tolerance for prediction error could inform this in the sense that like if you make sufficient changes to the room, basically your learned model is no longer telling you correct information about what this room looks like, and that might kind of push you to say like, okay, we just need to
Right,
re-anchor, we need to create
right,
a
right.
new map.
Just right. And I imagine again mentally, it's like if I was the rat, I'd say, "Oh shit, this isn't the same room. This is different." You know, where am I? Type of thing.
This
Right,
seems consistent with the idea that the voting ID is what re-anchors the grid cells, or like if the object ID changes, depending on maybe like some features that we've seen, and it pushed the object ID to change,
right.
then the re-anchoring of the grid cells would happen.
I hope you're right. Not. I mean, it's certainly moving in that direction, right? It's sort of saying, yes. I honestly, Rami, I can't. I haven't been able to internalize it myself, but it suggests alternate ways, like how to think about re-anchoring occurs and how it might occur over larger areas, and um um right, something
Yeah.
like that. It's uh anyway.
Because
It's
it's always
it's
tricky when you start with a model free, like who's then recognizing, I don't know, these big features and how is that informing object ID or
right.
um
So I was trying to trying to find papers. What do they know about this? Has anyone figured out a neural mechanism for how these environmental clues actually force the remapping? And I couldn't find good stuff. What I did find is that, and Hojae and I talked about this, is that place cells themselves are mapped from environmental clues. You don't need grid cells. And so, environmental clues will map place cells. And so like place cells and grid cells work independently, but then together. So if you have place cells without grid cells, you'll get a place map, but it's not necessarily as good, and you can't do path integration. If you have grid cells without place cells, the grid cells lose their grittiness and get confused. And so these two things work together. But it's all, how do the environmental clues determine this? Why only large scale distal clues make a difference and not close ones? I don't know, but it does, to me, it suggests that it's an area to explore, to say like, hey, what if reference frames work like this in the cortex, and then reference frames were essentially sort of sent to represent a class. Initially, it represent a class of objects that share basic shapes, um, and then you would start with that. And they wouldn't be this impacts all the things we talked about, like when do you start new reference frames, when you start new models and so on. Anyway, this is kind of an eye opener to me, that there could be the reference rate could be determined in a model free sort of deterministic way. The other thing that came out of the papers we were reading is that you need more than one grid cell module to do this, to get a good mapping. And so Hojae is going to talk about the simulations, but they were doing simulations with maybe 3 overlapping grid cell modules. So you have to have 3 modules that that remap independently, and that's sufficient to create a good representation of the space. And I didn't understand how that worked. And so Hojae looked at some of the models that they were doing to explain how do they write, what kind of model they do that says, hey, if you just have 3 grid cell modules, that's enough to map the entire space properly. I'm sorry, this is a poorly written up description of current thinking, but that's all I have to present here today. And maybe we could switch to Hojae to talk about how these models did that.
Okay, let me switch over. Oh, sorry. I was wondering where the earthquake
Oh,
was very high up here, just FYI.
well, Northern California.
Yeah, like we're all down here, so I think we're fine. Anyway, so I put my stuff in another X calendar, but I can move them over later if needed. So yeah, so the papers that I were looking at primarily were these two. One called modular realignment of entrinal grid cell activity as a basis for hippocampal remapping, and this one, it's a little bit small, functional independence of entrinal grid cell modules enable remapping in hippocampal place cells. And well, first of all, I think we need to um, clarify remapping versus realignment. So I think one of the reasons why these two papers exist was they both reference this paper very heavily in 2007, which showed that place cell remapping was accompanied by a coordinate shift in grid fields. So when we say remapping, place cell remapping, when a rat goes from like one environment to the other, you can get a completely new set of place cells, firing, and also the old place cells from the old environment. They can either be silent or it can be active in like a completely different location. So that's what I mean by remapping, or I think that's what a lot of papers refer to when they say remapping.
You're saying remapping only refers to place cells? Was that?
I think that's I've only seen it in the context of place cells like remapping. And then the grid cells would they would keep their structure, but they would change their patterns in some way. The hexagonal patterns of the grid cells can either, I don't know, shift, rotate, shear out, yeah, whatever
No,
that
no,
is.
I think there's only the 2 basic ones. There's which cell is active at some point, right? So that's like, um, That's just picking which is the active cell at some point. And then there's a rotation. So the whole grid cell module can be rotated some amount
Yes.
relative to the environment. There was one of these papers also looked at a third variable, which I thought they kind of dismissed, but sometimes the grid cell modules they're not orthogonal in every direction. They sort of like they get stretched in one direction. They were looking at that. But I think they dismissed that. So the two major ways that grid cells remap are rotation and which cell is actually active at any point in time at a location. I don't know what the right word for that is. I forget.
Phase shifting?
Phase shifting, right? It's confusing.
Yeah.
Uh,
Yeah, some of the terminology is indeed a little bit confusing. Right. So, and so the specific questions that these paper, at least this one was looking at, is if the grid fields are kind of, if they're, so basically there was like a little bit of tension between grid fields kind of changing in, I guess, predictable ways, and how that leads to place cells completely remapping, and how many modules of grid cells are needed to cause that hippocampal place cell remapping. Okay, so yeah, the puzzling question that I thought was happening in this paper, place cells can remap dramatically between environments, but grid cells, which are, I guess, I'm going to say one of many inputs of place cells now, because I feel like um, grid cells and place cells are very uh, interrelated, but I've also seen papers where, again, place cells activities can happen even if the grid cells are inactivated. So when I say it's one of
well, that's what I
many,
was saying. That's what I was saying earlier. Is like place cells seem to be driven by environmental clues.
yeah,
In addition to grid cells, which was confusing because these papers I think are just showing how grid cells can change the place cells.
yes,
Right. Right.
yes, yeah, yeah, yeah. So, um, yeah. So the question was,
Just before that, sorry, with the realign coherently, what does,
yes,
I guess, coherently mean in that context? So as in,
uh,
um,
yeah. So
I,
because I guess if they
coherently,
realign, if they all realign in the exact same way, then we wouldn't expect a different place cell.
I
right,
think
right.
I
Go
think
ahead, Jeff.
I think coherently means they're just saying that um, if I take the cells in a grid cell module, they all their relative position in the world always is the same. Like they like that you're not scrambling the grid cells. There's a set of them, and they represent a set of points in a sort of in a hexagon or a diagonal lattice, and that they all move together. That is two cells that are adjacent to each other in environment A will be adjacent to each other in environment B. It's just
Within a
that
particular grid module.
in a grid module,
Yes,
right? So grid
yes,
Right, yeah. So
modules,
so basically just saying grid modules, as we would hope or expect, don't break when
r-
during remapping.
right,
yes,
right, right. I mean,
Or like
so
don't do something catastrophic
this
and
was
weird.
that idea is from the very beginning of grid cells. Oh, there's a group of cells that work together like this, and but they constantly remind these papers that they constantly put words in this as if they're questioning things we already know. Yeah, so reline coherently just means that they act like grid cell modules. Right,
Yeah,
yes.
thanks.
Yeah. So yeah, my definition was coherently was that if there's a transformation like rotation or phase shift, it's going to be applied to the same for all the cells within a module, but there can be different transformations across modules, and somehow all of those combined together can cause place cell remapping. So there's actually a question of like, if we apply different transformations to each module, like how many modules we need to get remapping.
Right. So if you have three modules and you can only do phase and rotation, then you've only got six variables to work with. Three modules
Mhm.
each can have phase and rotation.
Yeah, I'm going to actually show a figure from this paper that was actually kind of informative of that three module thing. Let me see. Okay. Zoom screen. Okay, there we go. It's this one. Oh, sure. Oh, okay. Just pops over here. Okay, so let me see if there's anything from above. Oh, okay. So the methodology, this is a purely computational paper. There's no biological experiments, but the next paper, if I get to talk about it, it actually did simultaneously record hippocampal P cells and grid cells using neuropixels. So that actually like biologically shows that yes, they have observed hippocampal place cell remapping at the same time as the grid cell realigning in this rotation or phase shifty way. So, how they did the modeling. Go ahead, Jeff. Sorry.
I guess the thing that I was trying to get out of these papers was like at first it seemed this is almost like impossible. How can you have just, 3 grid cell modules with just, 2 variables in each one, somehow create this unique map. And so without any learning, I don't know, it just seemed a little mysterious to me. And I couldn't understand the networks they were modeling. They built these networks. So I asked Hojae to see if she could help illuminate what these models look like.
Yeah. Yeah. So so how the model set up. So there's several components. So there's a model for grid population. So all of this is just one equation to simulate the grid fields. So that equation that I just pointed out, like right here, g of x, blah, 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 is all that it's going to do is going to produce this hexagonal grid firing patterns that we know of for grid fields. There is also a place cell network model. So grid cell activity, it's again, the equation is g of x. So the place cell activity, I think R is some like firing rate of place cell, something like that. That's dependent on grid cells multiplied by some connectivity matrix W. And then some additional, these terms are just like additional like inhibition terms so that place cells will be actually, the activities of place cells are actually sparse at the end.
So is this connectivity matrix random initial values? Where's it come from?
Yes. So in this paper, this connectivity matrix is first of all sparse. So only 33% of the matrix is non-zero. So
It's not super sparse,
yeah, it's
but
not super sparse,
but it's not
right.
fully connected.
But it's not fully connected.
So what?
So,
So a grid cell would connect to 33% of the plate. I mean, a place cell would connect to a third of the grid cells. Is that
yes. So yeah, so in this model they had 1,000 grid cells simulated and 500 place cells. So each place cell would get inputs from about 333 grid cells because 33%. And then the weight in those 33% is just randomly initialized between 0 to 1, and it's kept there. It's not learned. It's fixed there.
Yeah.
Which is kind of weird. Yeah.
What does this learning rate refer to at the top of the page? Where you were like activity of dependent plasticity and recurrent excitation, where
yeah.
epsilon is the effective learning rate. So what is being learned?
Okay, so this is an additional activity that they did afterwards to see, instead of keeping the weights fixed, if they could actually change the weights by this, with the learning rate.
To achieve what result?
Well, so what they found is that, this gives basically better place cell place field maps. So like without changing any weights, the resulting place field maps were, it looked like this, but these are actually not super realistic. I think the authors were saying, okay, these are not like completely sparse, and also the place fields are actually smaller than what's biologically observed. So they said, but if we do some learning, it can
Well, funny
make
because I
place
look at
field
that
maps.
and that looks like place fields to me. I mean, you got like one, you know, a cell responds in one location or maybe two.
Right, right. I think
That's
it's pretty close. Yeah,
that's pretty typical what place cells look like. So
yeah,
um, I was this is key, I think, because I was under the impression that they got good mapping, that they were
yes.
able to, without any learning, that just with, well,
That is
assuming
my
enough.
conclusion as well.
Okay.
Yeah. So, I mean, they said you can have a little bit of improvement, but ultimately, I don't think learning was key to good place cell remapping. Like, you don't need to learn to get a good place map, and you don't need to learn to remap the place cells after you perturb the grid cell modules. Okay. So the connectivity is only it's there's just this one matrix in this paper. The other paper has a little bit more nuance to it, but I'll get to that.
How
But
many
anyway,
cells per how many cells per grid cell module? I mean, oh, there was 300. There was a
so
thousand. Wait a second. There's 1,000 grid cells,
yes, 1,000 grid cells.
and so there's three modules.
So here they tested different number of modules. So
Um,
there was a setting where it was like one module. So if assume that all 1,000 cells are in one module, that's not going to do remapping very well. Actually, let me just get to that point. So okay, so where it says S1, S2, S4, that's number of modules. So like with and the y-axis is like the degree of remapping. So the closer to one, there was like a complete remapping. If it's closer to zero, it's like, okay, there was some partial remapping. So if you assume that there was only one module, there's not a lot of remapping. But then if you have up till 16 modules, so each module will have equal. So here they equally divided. So,
they keep the
six,
same number of grid cells and just divide them into more or fewer
yeah,
modules.
divide, yeah, yeah. So same
Hmm.
1,000 grid cells, but if you increase the number of modules, the number of grid cells inside that module is going to decrease. So if it's like four, I guess it's like 250 cells per module, and four
Hmm.
module will be like 1,000 cells, right?
Hmm.
So that's where we get this. So,
And then, oh, sorry, and then
yes,
they, in those conditions, they do only shift or only rescaling or only ellipticity.
yes, that's correct. They have not tested like interactions between
The
these two.
combinations.
Oh,
Yeah.
Because
interesting.
it looks like with only shift the one module, it's already more than 50, 0.5, whatever that means. I'm just,
Yes.
yeah,
Yeah.
curious how it would be if you did one module with shift, ellipticity, and rescaling. But probably it wouldn't get much better because,
It just
I
you
guess
just
one
can't.
module is also kind of weird because one module, then there's like
yeah.
1,000 cells that all kind of fire almost at
Yeah.
unique locations.
Yeah, they're like little grandmother cells or something, and it was like,
Yeah,
Yeah, I
it's
mean,
almost
it's
like
Yeah.
a yeah.
just surprising
Uh,
that it's still at over zero point 0.5. And
I guess it kind of
yeah.
depends maybe on the scale and how what the scale was for that grid cell module versus the environment. Because
Yeah.
if
Let
it's
me get to that.
yeah,
Yeah. So hold on, let me also just double check. Okay, so I think the caveat here that you need to recognize, like I think it's high, but it's probably not actually as high as we think because the way that I define grid cells, they sample the sampling between grid cells to be, between the spacing to be like, 30 centimeters, 90 centimeters. So within a module, right, this was a paper like 2011. This was before the 2012 paper that biologically showed that grid cell modules are within a module, they have similar spacing. So what they did is that they like one module can actually have grid cells whose spacings can be anywhere between 30 to 90. Does that
oh,
make sense?
oh my gosh, so they're not uniformly mapping the space.
Yeah, they're not uniformly mapped from the space. So
Oh,
that's the random modules. Just hold on a second there. That's the random modules. The frequency modules, they sort it from lowest, after sampling from 30 to 90. If you have two frequency modules, they sort the spacing. So the lowest 500 will be in module one, and then the higher 500 will be in module two.
I'm saying, what's the frequency in this case?
frequency was spatial
Oh,
distance. So
oh, I
the
see.
modules,
I
instead of
see.
being randomly assigned, they sort the grid cells by the spacing, and then the smallest
All right.
spacings will be module
So
one.
they start off
but
with
you
a,
can still have different scales or frequencies within one module.
Yes, you can still have different ones within a module. Yes,
It's just not totally random across modules.
yes, that's
that's
correct.
weird. So they basically
Yeah,
started with 1,000 grid cells, have varying scales, and then they randomly grouped them together into modules,
yes, yeah.
and then they started not randomly grouping them. Oh, that's weird.
Yeah. So what's closer biologically now is you have to look at the frequency modules. And this one, you can see that it needs much more number of, well, not, I mean, it can still remap, but basically you need at least four to have, these remapping or turnover values. Um, the more the better.
So you're
So
saying grid cell modules that look like the ones we're familiar with
yes,
take more.
yes,
Make that
yeah,
which makes sense, I guess.
yeah,
But and how many? If four is not that number in there is the number of modules.
yes,
So
yeah.
even
It's kind of
So
four.
interesting though, because what if in Cortex it was more like the random one? Like you have kind of one module, but each grid cell almost has like a different phase
I don't think that's,
or frequency. Because
well,
But that but I mean the problem with that is I mean that's I think would probably fly in the against the way people think grid cell modules work, right? I mean,
yeah,
definitely in entorhinal cortex.
right, but we have to have some kind of mechanism that works in cortex, and so I've always as a first approach said, hey, it's probably going to start with what we have in entorhinal cortex, and then you can vary it a bit.
right.
Um, I don't know. That seems weird. I don't even know how you do path integration
Right.
if the cells have all the different scales to them.
Yeah.
It would be weird. I don't know.
Uh, anyway, so but that's a caveat for this paper. So the newer paper, the Lykken, I'm not sure if I'm pronouncing correctly, 2025. I'm sorry if you're listening by some miracle. Um, this one actually does have more biologically realistic grid cell modules. It only has three, and each one has uniform spacing. So, anyways, so let me get to that part. Okay, so this paper was also the one that Jeff posted a few questions on, and it's understandable because this paper actually doesn't do the modeling. It refers to another older paper. Sadly, you look at this one that actually provides the model. So
Oh,
yeah, so that's why it's like, where's all the details? It's like there's no details because it's just in this other paper. um, okay, so this paper again, it's another kind of model for single place fields of CA3 cells, a two-stage transformation from grid cells. And they come up with a similar, similar model, but that goes from grid cells to at least CA3 place cells. And so instead of a direct grid cell to place cell model, so instead of having like one weight matrix, they're more closely aligned to the if you guys remember from like I don't know a long time ago, like the trisynaptic pathway from the entorhinal cortex to at least CA1.
mm.
So like they have grid cells, but they also have included like 200 granule cells or something like that. It's a little bit weird. um, yeah. So like if I had to make like a coarse biological analogy, the Monaco and Abbott paper was, grid cell to place cell directly. It's like more similar to monosynaptic, and this one is like closer to trisynaptic. So they're kind of two different ish models. Um,
And what does that effectively mean? Like, do those, um, what were they? Granule cells have like some other input, independent of the grid cells, or is it more just like kind of expanding the depth of the network almost?
I
Intermediate layers.
yeah. Literally, like instead of a one layer neural network, this is like a two. There are two stages, basically two layer network.
Yeah. Okay.
Yes, so I don't think it's
But
that
it's not
different
like
actually.
a
Yeah.
crazy new competition, like I don't know, that gets some additional movement or sensory
No,
input or
no,
something like that.
no, no,
Okay.
no. Nothing as complicated as that.
Well, they probably have no idea what the hell these cells are doing. You
Yeah,
know,
that's correct.
I mean, every one of these cells is like thousands of synapses or hundreds or whatever, and you know,
Right.
it's really it's wild
Yeah,
territory out there.
but at least the model I think is like a little bit more biologically realistic in the sense that so again, the difference between the two, this they have more grid. I don't know if this number is realistic actually, but this is a number that they used.
So
They kept
so
it
what?
to
10,000 grid cells. Is
and
that again?
200.
Is that are they going to do the same thing where they take those 10,000 grid cells and divide them up into some number of modules? Is that what they're
Yep,
doing?
yep,
Okay.
yep, yep. And 10,000 grid cells, a few dentate gyres, I don't know what is it,
Hmm.
granule, whatever. Um, Divide into 3 modules of different sizes, so it's not all uniform size like the Monaco and Abbott. So M1 is the largest, M2 smaller, and M3 the smallest. And I think
Yeah. What was it? Yeah. Go ahead. Justification for that.
the justification was that the cells near the dorsal side
Hmm.
um, was
They
contributing
rep
more. Yeah, so they just put more cells there.
well. I don't know. That's weird. I mean, all right. I mean, when you're on the dorsal side, you have small. I think it's a smaller scale. When you're on the ventral side, you have the larger scale. I hope I got that right.
Yes, that's right.
Uh, and then, but I had no. I never heard any evidence that the grid cell modules have different number of cells in them, and so they just make that up. Um,
Uh, yes, but at least
okay.
proportion wise, if you put more cells, out of 10,000, it will have a little bit. It will have more contribution just by the number. I'm not
But
sure
yeah,
that
it does feel
that's
like just changing the weights be a more natural, like the
right.
synaptic strength would be a more
Right.
natural way of doing it
Right.
if that was the main
Yeah.
reason.
And also, the fixed spacing between each of the modules. So, again, the dorsal side or the M1 it has the smallest between like 50 centimeters and the
Well,
larger one is
this gets really weird. It's just really weird. I mean, if I have a lot more cells in a module, does that mean that they repeat less often? I have to go much further. Are they trying to make the repeat spacing the same in these different modules, which is not right? That's weird. That's really weird. I don't get that. I don't know why they're doing that.
yeah. Um, yeah, but otherwise, sparsity is about 14%, I guess.
Well, sparsity of what?
Grid cell to place cell. So each place cell, they said will get
Oh, the
inputs from 14.
connectivity.
Yeah, the connectivity. Sorry.
Mm.
Yeah.
But would those fire the
Um,
DG cells? Because I thought there was only like 200 of those.
Yeah.
uh, it's a little bit complicated.
Which made it kind of sound like almost like an autoencoder, like it was kind of compressing it into like a lower dimensionality.
Yeah, so it's not just like all the grid cells innervate the granules, and only the granules innervate the the place
Okay,
cells.
so it's
It's
They
both.
had
it's
they had
both
both
of them.
pathways.
Yeah, that
Okay,
both.
okay, then that makes
Yeah,
sense. Thanks.
yeah.
Yeah, but
Um,
what the hell's what the hell the granule cells doing? They just throw them in for good measure? I mean, it's
y-
like what?
I
What?
think it's what I said, but you just said it out loud.
I
I
mean,
said inside yesterday.
okay. I mean, then all of a sudden you really have no idea what the hell the network's doing. So if you, I don't know, at least I don't.
Right. But
Okay, this
yeah,
is what
but
they did. This is what they did.
yeah, this is what
All
they
right.
did. And so this paper, honestly, it's not a remapping paper. It just shows that yes, with this setup, you can get reasonable place field maps. Um, and what the Lykken paper did is it took that model and it tried to do the phase shift and the rotation, and found that rotations under this model does not cause remapping, but the phase shift does.
Really?
Or rather,
Yeah.
rotation just doesn't happen.
And
Like
no
if
rotation.
rotation occurred, it could contribute to remapping, but they just didn't observe rotation happening.
No. They in the simulation they apply the rotation transformation that did not lead to the resulting so now that we have perturbed the grid.
Did they remap? Did they rotate all the modules or rotate them independently?
They rotated them independently for each of the 3 modules.
That's hard to believe that it wouldn't lead to remapping. It's just so it's so weird. It's hard to imagine because it means it's coincident. The grid cells that are coincidentally active would be changing, and so you wouldn't have the same grid cells active together. It's weird. I don't
Yeah.
I don't get that.
Okay, so let me see if we can see any partial remapping.
Yeah, like if it's totally invariant to rotation, that's kind of weird.
I
Unless
don't.
they rotated
Yeah.
specifically by like 60 degrees or something like
Yeah,
that, and
but
it
even
fell
that,
on
Right.
the hexagon. But yeah, even
yeah,
that would
but
probably
even that would be weird. I mean, we're rotating the grid cell module relative to the environment. And
right.
so the cells would be they'd be firing in different the grid cells would be flying in different places. Um,
Oh, okay. There's like 91 pages. It's gonna be hard to scroll. Okay, I'm just trying to see if I'm just trying to get to the figure section. Goodness.
But yeah, these are the modeling results they had. Because yeah, you said it was like a mixed experimental and modeling paper.
Yeah. So, model this. And also, I think they have observed this biologically. I think they have, because they were recording from both place cells and grid cells. Let me actually, well, let's see. So that's clear. Is this any? Okay, but all right, never mind. I don't think I can actually find the right one. But let's see, where am I?
I mean, I guess the other paper the first paper you showed, also the results seem to show that rotating doesn't have as much of an effect as. Oh no, they have rescaling. They didn't even test rotating.
Yeah, let me see. I thought they, yeah, shift. Yeah, they didn't even test rotation.
Okay, never mind then.
Because yeah,
wow,
just
I didn't
at
know
least
that.
looking at
I
the
didn't catch it.
abstract of Lykken 2025,
Mm-hmm.
uh,
Mm-hmm.
hippocampal remapping was not associated with rotational differences between grid modules under any condition. Um,
Yes, yeah.
so that sounds like at least that could be referring to an experimental result that they just didn't observe it.
And I just can't imagine how that would work.
But like I could imagine the grid cell modules just don't rotate during remapping.
Oh, but okay, but I thought
Like
that's been observed. That idea came about from I think original observations of grid cells, I thought. Um, in fact, I remember there's a lot of data about how they rotate. There was they didn't rotate equally, and you can only rotate up to 60 degrees, and then suddenly it doesn't mean anything anymore, or something like that. And then, but there was statistics on what the actual rotations observed were. Um, so they've been observed to rotate. I'm sure of that. I mean, at least over papers, unless those papers have been discounted now.
I guess I'll
Um,
double check if the rotation was truly independent across all 3 modules. If not, then I can see why it wouldn't lead to remapping. Like if all 3 modules were rotated by
right,
12
right.
degrees, I don't know, then that's probably too coherent to cause remapping. Um,
Right.
but yeah.
I mean,
Yeah.
let's pop up against here. I mean, none of this is going to end up in Monty. Um, the reason we're looking at this is trying to understand just different conceptual ways to think about reference frames. And so it was interesting. I think it was interesting to show that a few number of grid cell modules can lead to complete remapping of location spaces.
Hmm.
There's a lot of details that were confused by that. Um, I thought the observation made earlier that these can be initially model free or without learning, and they work pretty well. And then I think what I've read is that the place cell locations can, if through they're an animal goes in an environment, there are place cells and there are grid cells, and navigation works. But over time, as the animal gets more and more familiar with that environment, the place cells, there can be more place cells added. That is, especially around areas that are of high importance to the animal, and then the firing rates can change and so on, but it doesn't get remapped, you know. So the idea that there's this sort of non-model based ability to establish reference frames based on large distal clues of environmental clues is a way to think of that, how we might want to think differently about grid cells or about reference frames in Monty. I don't know I don't know what the answer is yet, but you know, Rami, you pointed out earlier, like, oh yeah, maybe it might make it easier for us everyone to lock onto the same object because everyone has just you know, there's all the grid cell modules have to lock together.
Yeah, and I guess an interesting question is sort of the almost like the chicken and the egg of how the place cells versus the grid cell sort of alignment
Right.
is determined in the first place. Um, because like
Well,
a lot of
back
these papers were focused on showing that like, okay, yeah, you different grid cell activity with random weights can result in different place cells. But what we almost want is more like, okay, given these sensory inputs, that determines the place cells, which tells us how the grid cell
right,
should anchor.
right. But again, in this case, it's the large scale sensory inputs, which is a clue, but I don't know what to make of that clue yet. Um, you know, the way I
I mean,
keep thinking about
I
it is like
think
the overall,
yeah,
it's the overall shape of an object. It's, you know, like a proto shape or something like that, but unlearned, you know,
yeah.
Yeah.
weird.
I mean, this doesn't really help us, but because it relies on cortex. But I seem to remember what is it? I guess the lateral entorhinal cortex. I think, which is kind of the more sensory input. I think that receives input from sort of high level visual cortex. So you can imagine that sort of receiving input from large scale objects, maybe
Mm.
um, sort of high in the hierarchy. Um,
That goes to um that goes to MEC. You're saying what are those?
LEC, I think,
LSC.
uh,
Great, great.
which I think is the more kind of sort of sensory or feature part of the of
Mm.
entorhinal cortex.
Yeah,
Um,
right. Mm. It's confusing.
but then,
Um,
yeah, how that would map onto like a cortical column, other than just saying, yeah, it's I don't know, inputs either from other columns or direct sensory input.
right. Well, we can imagine how somehow in Monty we would say, okay, we're gonna try to establish some sort of reference frames that are um, that can be established very quickly from some sort of large scale clues. I don't know how that's going to work yet. Um, and then we work at refining those over time. Um, I don't know. We're at the state. We're still in the stage of this exploration where everything's very confusing and we don't have a clear concept yet. But that doesn't scare me. I'll have to keep going at it. What's this, OJ?
Um, well, a couple, I guess my speculations, I guess, about just extending on your post from Slack about some
Yeah.
speculative thoughts. So you said place cells are a randomization specification function that takes low-res grid cells and convert to a more proper SCR. This allows the system to work with only 2 or 3 grid cell modules. Um, I think I agree with that now that the place cells are better candidates to represent the location SCR than the grid cells. But the grid cells are responsible for it's still like the SCRs coming from grid cells is just the SCR represented by place cells are just readouts, I guess, of the underlying grid cell
Yeah. You know,
modules.
the place cells themselves aren't like a location SDR. You know, they see me as this cell fires at this location. And if you really want to have a location representation, you'd have to have a lot more cells. So I was imagining, oh, what if there's a lot more place cells and they only show some of them? I don't know. But
Yeah.
anyway, That's just a secondary thought. So
Yeah.
these are right. I want to point out these are things that these were not conclusions. These are almost like questions. It's like,
Right.
is
Well,
this the right way to
yeah.
think about? Is this the right way to think about this?
Yeah.
But yeah, I
And
guess
then
it relates, I think, to something we were talking about the other week, which is that kind of even if the grid cell code isn't that unique, the sort of intersection with sensory features, say, could still define quite like could basically enable the whole system to work. So
right,
as
right.
in like a certain set of grid cells, like this cell in this module, this cell in this module, this phase in this module, whatever, those could all be active for multiple objects. Um, but then they will predict different sensory features. And then within those sensory features, within those mini columns, we would get a different cell that's selected as the winner.
Right, right.
And then that
Yeah.
recurrence could kind of ultimately still lead to kind of a stable, unique object ID emerging.
Yeah.
But they're basically there would just be a lot more interference between the different kind of encoding spaces in the reference frames. But it doesn't seem like it would maybe fundamentally break.
Yeah.
Yeah. Um, I like so, and then there was like the other potentially how it could work in the cortex that you wrote, Jeff. That I liked at least. There are grid cells equivalents in L6A. These projects to L4 and depolarize a set of L4 cells through fixed connections. The depolarized L4 cells are like place cells, but not active. There would be enough depolarized L4 cells such that most mini columns would have one. When a sensory input activates a set of mini columns, the cells to learn will be chosen from depolarized cells, not randomly as we currently assumed. Um,
This again was a hypothetical saying, well, it could
right,
work like
right.
this.
Yeah,
Um, I don't know if everyone followed that. That's a really complex sentence. Um, um, I'll describe it again. Right now, when we pick cells from in layer four, we randomly pick the cells in the mini column to be active. This was saying like, what if the grid cells project? We had several grid cell modules overlapping in layer 6A, and they projected to layer four, and they in in a sort of a deterministic non-learned way, at least initially, and it would depolarize a whole bunch of cells in layer four. So a particular grid cell location in the environment would depolarize a set of cells in layer four deterministically, like just that these are the ones that you can use. And then a sensor, and that would be across all mini columns. And then a sensory input comes in, and you would say, "Oh, well, pick the ones that match up with uh, the that were depolarized from the location." So in some sense, the location says these are the cells you can use. I'm trying to do this all in a sort of a non-learned way.
yeah,
Very speculative. So if you
yeah.
didn't follow that, don't worry about it.
I feel like it works. So,
Okay,
well,
I'm
but then
glad
I
you
need
like.
to, yeah. Well,
I'm glad you
I need
liked
to
it.
remember like why. Like I was trying to remember why. Like, oh wait, this idea couldn't work. I guess. So, anyways, so, like,
Well,
this is my
I guess it's the question
rephrasing.
of what direction do we want. Again, it goes back to like if we want like large scale visual features to sort of define the grid cell space,
Mm,
then it kind of feels like we want like the same thing to happen, but sort of in the other direction.
Yeah, but that's weird because we want these large scale features to define the space, but then we want to be able to learn the local features that are small in that reference
Right.
frame. It's
yeah.
like it's
Yeah, so
so
maybe
weird.
so that maybe that has nothing to do with L4. And then the question is like, okay, is that like top-down feedback?
Well, I was thinking
Or
L4 is learning the local features, and
yeah,
yet somehow the grid cells um got anchored not by the sensory input to L4, but by something else.
Yeah,
yeah,
that could be the layer 2-3 object ID, maybe.
Right,
Which, yeah, maybe there's, this goes back to the general versus specific category ID, where
right,
maybe there's some input to the higher layers that just tells you the rough shape. It could even be to, I don't know, layer one feedback connections or something like, or layer six feedback connections that initialize the reference frame. But yeah, something that basically represents a category of shapes.
right, 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 right.
But then,
Yeah.
yeah, that doesn't work. Just think about it, because
Well, I
then
mean,
we
I
just have
think
a one, like we still want to have a unique reference frame for every unique object, so couldn't have a category
right.
reference frame.
I don't know. I don't know. I mean, the right answer is sort of some intersection of all the crazy ideas we're talking about. So it's, it's not, we're- yeah, I don't know.
Yeah.
That sounds right until it doesn't sound right. And everything sounds right until it doesn't sound right.
Yeah, like
Mm,
relying on top-down feedback is kind of weird
Right.
if we generally assume that like
yeah.
learning, well, I guess learning happens slower in the lower levels, but if the top is the hippocampus, that could be where learning happens first. So
Yeah.
if like if the hippocampus is almost recognizing objects, it could be more like object
Well,
shapes.
we do know that the hippocampal complex recognizes large scale environments. So, um,
But then it's confusing.
yeah,
It's like, well, how would it do that without cortex if we've not learned models yet?
I don't know. I
And
mean,
then
I
it's like
think,
some
Yes.
crazy
well,
bootstrapping
this
thing.
seems to be the right. This is, but the key insight that's driving all this discussion is that um, this idea that maybe the reference names are representing these sort of non-learned prototypical shapes, and or not even prototypical shapes. They're just it's some hashing function of the features that says, yeah, this shape can be represented roughly, and then that's what the reference names represent, as opposed to it's more of like, as we were just saying, it's more like the class of objects. All the objects that have this basic shape have the same reference name, and we keep bumping into that over and over again. That idea. So this pushes me more in that direction. It pushes me to say, well, hey, maybe that really is the thing about reference names. They're more prototypical or generic shapes, and then we do refinements on them somehow. That's what would be implied by this. What's your last point here, Hojae's mental model?
Oh, yeah. Which is just more or less, in my words, what I would say the idea was that we can imagine there's some overlapping grid cell modules in L6A with different, I called it phases. So how it could work basically, and then we will still get like movement information, like displacement information to L6A that can change the grid cell modules. So path integration can still happen. And then with the fixed connections, we'll still have places like SCRs. I thought in L4. So another way to state that is updated grid state deploys as some subset of L4 cells. Place cell is just SDR readout of the updated grid
Mm.
state.
So
Again,
this is a rephrasing of the longer paragraphs above. Is that right?
yeah. And then features activate the mini columns, and this cells are deploys win and learn. So it's a
Hmm.
sensory, yeah, gives the mini column.
And just to clarify, what you say in L6 with different frequencies, right? As in like different scales,
Grid
rather
cell
than different
modules
phases.
was different. I mean,
Because
whatever
different
the
phases would be like the phase would change within a grid cell module.
yeah.
Um,
So I mean, I'm using the language from the paper, which was like phase shift. Like, so I guess
But yeah, well, it's funny because in the entorhinal cortex, you have this steady movement from large scale to small scale in this one dimension, right? This
yes,
dorsal ventral dimension.
yes,
And it's like a linear strip, like I showed last week.
yeah,
In the cortex, you have this two-dimensional sheet of cells and all these modules next to each other, these cortical columns next to each other. If there were overlapping grid cell modules, it's not clear there'd be changing in there'd be any sort of uniform like change. Like in the fovea, they'd all be small, and the periphery, in V1, they'd be bigger. But it's not clear that'd be a steady, I don't know, some sort of steady march as you move out from the center of the uh, V1 to the edges. So
yeah.
the different grid cell modules might have the same spacing. Um, I don't
Mm,
but
know.
I guess then going back, then I mean that just makes it hard. Then I guess to get the unique code right if they're also not rotating. Um,
Uh, yeah, I don't know if I believe that, not rotating, but um,
yeah, I guess we need
they could
we need some
they could
way for
have
them to be
they could have
uh
slightly different spacings. I know, but I just don't think it would be like when in MEC you see this like
Right. Huge
1.4.
scale differences.
Every
Right.
grid cell model is
Yeah.
1.4 over the next one, and square root of 2.
Yeah.
And that I don't that's not just you can't do that in the cortex. You'd be by the time
Right.
you get to the
Right.
outside, you go from the center of V1 to the edge of V1, you'd be representing the universe or something. So it's like um
Um,
I don't know.
so yeah, I
So
mean, I'm not saying that the grid cells are set up the exact same way as EC. I'm just saying that there's at least different ones that are that can be combined to remap,
right,
to create enough capacity, basically. Because I don't think one is enough. So
right.
three,
Uh,
maybe there's different spacing, organized in some way, but in
I
whatever
think
way that produces, yeah,
and just in case anyone's scared about this, our goal would be to make as few changes to Mani as possible. So it's not like we're going to implement all this stuff. None of this is going to get implemented. We're just trying to understand some basic principles that we might think again, like, oh, the reference frame is representing a proto object and we need class IDs or something like that. Um, and there are ways that we can justify synchronizing a whole bunch of columns together, or how we might think about how they all know the location on the object that's being learned, something like that.
yeah,
Um, because
yeah. Then
if you don't know the neuroscience, this stuff can be super scary and just like, what the hell is everyone talking about? Um, So don't worry, you won't, not everyone has to understand this in the end. All right, are we done with that? I wish we had more progress, but like,
yeah, I guess I mean this was like all very like literature grid cell phase, but I mean again the more interesting thing is like assuming this can happen, or the speculative thoughts are true, like what does that mean for shared learning, or what does that mean for
right,
voting, like
right.
that part. um uh, yeah, like maybe we can, I don't know if Jeff, you
It would be,
continue to think about
in
this
some
more.
ideal world, I'd be able to put some signal across layer one, as Vivian was talking about. And that signal will
Right.
tell all the columns, oh, go to anchor your reference frame this way, go to this location, and this is what you're supposed to learn. Right? That would be
Yeah.
some dream world. Um, and these are clues that, hey, you might be able to go in somewhat in that direction. I don't know.
Yeah.
When things are not learned and you can anchor without learning, then it gives you this ability to anchor everybody the same way, because I don't know, it just makes it easier to do that. Okay, more work to be done.
Yeah, some completion. But it was, I was surprised that there could be no learning and still get like reasonable place
Me,
maps.
me too. Me too.
Yeah.
That was a surprising thing.
Yeah.
Yeah. I guess, yeah, just briefly on the same topic. I had this weird, like, finding a lost book in a library moment earlier where I was filing away some papers on grid cells in my Zotero, and then looking at all the grid cell papers I had there, and found a paper I never got the chance to read, but then it looked really interesting, and I was just curious if any of you have read it yet, because it's a 2021 paper, and I think the preprint already came out 2019, but I haven't seen anything similar since then. So, I
Yeah,
mean, it's
we
almost kind
referenced
of too
this
good
one
to be
in
true. We went over this one?
well, we reference it in the TBT 2.0 paper, and I think it's even
Oh,
referenced in the 2019 papers.
okay,
What's
that's
remind
probably why I got it in the highlight.
remind me what it's about?
It's basically these researchers that recorded in yeah, behaving or foraging rats in primary somatosensory cortex. And yeah, they basically say, to our surprise, we were able to detect the full complete complement of spatially selective firing fields, namely place cells, head direction cells, boundary vector border cells, grid cells, and conjunctive cells in the somatosensory cortex. And
I
so,
wonder if these are Chinese guys. Are they in China?
yeah,
I wonder if this is the team that wrote to me. Remember, I told you after um,
Yeah.
sometime
Yeah, I
in
Yeah, I
the
think we
remember
were
past,
talking
that.
about this the other day.
right? They
Yeah.
wrote and they said they found this in V1 and I think also S1, and they said no one believed them and they're having trouble getting it published. Um, I wonder if it's the same team.
yeah, because
Uh,
it
Yeah,
seems
no, I
like it's
think it
published
is.
in Nature now, but or Nature
this is
Cell
Nature.
Research.
Cell
Yeah,
Research.
but it's just surprising that since 2019, no, I mean, the technology would be there to measure this by other groups that, like, I tried to find other papers that look into this and couldn't find more. But anyways, they had an interesting breakdown and supplementary figure of the layers in which they found these cells.
Hmm. That's
But I haven't
interesting.
had a chance yet to look at the details of how they did
Place
this
Yeah, I
looks
collectively.
I
like
seem to remember the place cells weirdly were kind of in deep cells, and then the grid
Right.
cells were a bit more uniform.
Yeah, and I think, yeah, unfortunately, I didn't have time to read the whole paper, but I think they actually didn't look in the upper layers. But yeah, still that there are place cells in layer 5 and 6 is a bit
well,
surprising.
they're showing them spanning 5 and 6 for all of them. Yeah.
Yeah.
Well, that was an alternate hypothesis I had. Remember, we talked about just a moment ago how layer 4 could be depolarized. The other hypothesis, you could say, okay, well, the place cells are down, and I wrote this in my Slack post. They could be down, low, down in the lower layers, and that they project to layer 4. The place cells could act like our location signal, and if you had enough of them, if there's enough place cells, then they would be like an SDR of location. And then they could project a layer 4. So that was this sort of suggests more in that range. Um,
Yeah.
Yeah, and I guess with this paper, I seem to remember there was some stuff that was kind of it was hard to see exactly how it fits in that like I think the grid cell responses was so relative to the environment more than like what we would think of as kind of like a object centric reference frame. So it was
well,
almost yeah
but
it
that's
was yeah it was similar
with
to kind
these.
of the grid cells that you would see in entorhinal cortex, which um so I think at least some
I
people
mean,
thought this
that
is like a downstream
yeah,
um
but I don't.
input
I
from that.
think that's more of an interesting clue is to. I mean, and that's what they told me they saw in V1 as well. They were literally mapping the environment, and um and I'm not sure if that's inconsistent with what our theory is. I mean, essentially, why wouldn't V1 or S1 be able to map the environment if that's what the animal's attending at
Right,
this
and I'm
moment
not
in
saying
time?
it's inconsistent. Yeah, I'm just saying it's not the golden smoking gun of like but I mean, and we talked about this in the TPT 2.0 paper. There's various reasons it's much harder to observe it in an
Right,
object-centric reference frame. Like just things like you need to record these grid cells for a long time in order to extract these properties. And generally, an animal doesn't like interact with a nut or something long enough to record what might be kind of grid cells for that object, whereas they're in the same environment for potentially hours. Um,
right.
and just things like that. And yeah, and there was like one other interesting thing I think in this where they trimmed the rat's whiskers, and that did not affect the response properties of the grid cells. But I think
Yeah.
that kind of fits with it being a reference frame for the environment, where it's like it's going
Right.
to care more about the animal running than what the whiskers are doing. But then presumably, or at least we would predict that you would have some other grid cells that were sensitive to whisker trimming. But again, they're just harder to record.
Right. We don't know how much of the world the rat learns details and how much of it's just spending figuring out where it is in the world, you know.
Right. But yeah, because I think it was only last year. Sorry, I was just saying it was only
it
last year
takes
there was some paper where they found like a small ring-like, I think, grid cell responses for like an object within the environment, where they basically
hmm,
showed that like I'm forgetting the details, but basically like the rat's like running around the environment, and so the grid cells are kind of mapped at a certain scale, and then it's running around this small object, and they remapped to sort of this smaller scale map
oh,
for that
interesting.
object. I mean, I think this was all in kind of hippocampal complex, but
Yeah.
there was some like technological innovation that enabled them to do that, despite the fact that the rat would only interact with that briefly before kind of
Hmm,
deciding to move on. And it just kind of shows that the basically the smaller the scale, the thing that you're interacting with, the harder it is to record grid cells,
interesting. Right. Right. That's
just the
fascinating.
behavioral effects.
That's fascinating.
Yeah.
But yeah, it is unfortunate that there's no other like I don't know
Yeah.
follow-up studies.
Yeah, I might give this
And
paper a closer read to see if there are any more clues in there on how grid
Yeah,
cells might be
I'd
working
like to
in
read it.
neocortex.
Did you can you post that again in Slack or something like that?
Yeah, I think unfortunately they didn't look at like grid cell modules or any kind of more in-depth analysis of the grid cell part of it, but yeah,
No, just this
maybe.
the different locations for the different cell types is interesting.
Yeah,
yeah, and
that's
the
the
V1.
supplementary figures at the very end. So yeah, you'll have to click that.
Oh, okay. Well,
And yeah, the V1 paper they did as a follow-up. I think that's just on bioRxiv. I don't think that was ever accepted,
oh,
but that
so
was
so
a later one, I think.
so they did do that. Oh, interesting.
Nils, can you post the follow-up in the
Yeah,
visual cortex one? Yeah, because I think I mean, this
Oh
one's
yeah,
just
sure.
somatosensory, right? S1.
Yeah,
yeah.
it's called, yeah, I'll share it. It's called a compact spatial map in V2 visual cortex.
Okay, okay. Yeah, 'cause I read this one,
Um,
the S1, but
So Vivian just posted in the research the somatosensory one.
yeah. Yeah, so somatosensory one.
and then what I can also post, there was a short commentary that was quite useful on that first long paper. I'll share that. It's called a spatial map out of place.
It's like this is an error. Doesn't belong here. That's funny.
Yeah, it can summarize some of the stuff.
It's funny when you Google for all these topics around grid cells and place cells, especially if you look for neocortex, all you find is like Numenta's stuff. Our research meetings, meeting recordings, HGM school, discourse posts, and like
All
Numenta
right.
papers.
Yeah.
Either we're out ahead of everyone or lunatics. Which one is it?
Yeah, so well, you can't ask AI if there's grid-like cells in the cortex because they just cite us.
Right,
Yeah, it's
right.
like you're in an echo chamber. You're like,
I
Yeah.
know.
So that's why this kind of paper
it's
is
been
like,
postulated.
it is useful and necessary for somebody else to actually be like, oh, we found them, we think.
Ignore all previous publications.
Ignore all, yeah, ignore
But yeah,
previous publication.
if anyone's
Okay.
curious, also if anyone's watching this, so yeah, we uh, just had a paper published or accepted for publication called the TBT 2.0. But in the end of it, there's a box where we talk about basically, okay, that prediction dates back to 2019, even 2017. Why have there not been more papers kind of showing evidence of that? And basically, what are the kind of various challenges to because it's still a falsifiable kind of prediction, but yeah, it's just hard
Yeah.
to consistently measure. But
And
the good news, I guess, is the technology is moving in the right direction. Everything from being able to do kind of kind of scanning microscopy at deeper layers, at high temporal resolutions, in an awake behaving animal, all this kind of stuff is sort of moving in the right direction of being able to actually check.
yeah, but then researchers have to want to look for it too because it's sort of outside their standard paradigms, and
Yeah.
Right.
you know.